My name is Nik Sharma. I'm CEO and co-founder of BioCorteX. I'm sort of joined by my co-founder, Mo. If anybody saw the SpaceX launch approximately 2 days ago, just as the conference started, lots of the tech that we're actually using to answer this seemingly impossible question of whether this drug is actually going to work for an individual actually comes from that same basis. So ultimately, the question that we're all really concerned about from a pharmaceutical perspective is this drug going to work for me? Now, me could be the patient, the individual, the family member, or actually the pharmaceutical company. This is the largest lever in pharma. The annual spend on R&D is, it's astronomical. So, 244 billion on a global spend, and 139 billion on the top 20 pharma. But the success rate is only 4%. Now that's shockingly bad, really, when you look at it. So if you look at what happens in aerospace, if you look at an engine program is actually very, very similar to designing a drug. It's about the same cost in the region of 2 to 3 billion. You have the same, 100, 150 very clever people working on the problem. But obviously, we all get on planes and they very rarely fail. In fact, they always typically succeed. Obviously, there are sort of cases where that hasn't happened. But the way they're able to achieve that is actually by testing in silico. They're able to practice and refine everything computationally and fail in silico before they go out and actually put that engine onto a plane. Now, this isn't because the engines are, we understand engines. And so, just to say that sort of Mo's background is that he was head of AI at Rolls-Royce, the aerospace company. So this is something that, I sort of learned as an MD, PhD, was that we don't understand all of the physics actually behind engine design. And what we're doing is actually bringing that right first time approach into drug development here. And so the aim is to increase the success in sort of clinical trials by uncovering the drug-bacterial interaction. Now, this isn't something that's new. So this isn't a new technique. This is something that we're just late to the party on. This is something that we've co-evolved with bacteria. Every single pharmaceutical that's been approved or anything that any of you take may have an interaction with the bacteria. Now, that interaction can occur at different levels. So it sort of can occur in the gut, it can occur, it has interactions with the host physiology, and sort of finally, if, in tumors themselves, there are actually bacteria that actually can metabolize the drug. And that's something that's been known about for over 100 years. So what our tech does, our tech is very different. Now, there's a lot of hype about AI. Our tech is not a black box. It's a foundational emulator. Now, an emulator, if anybody had a misspent childhood and played a lot on computer games like I did, you can still get the same software for your Atari and sort of run it on your MacBook. Because what it does is that it emulates the hardware, and then you can just run the software on this. And this is something that NASA's been using, for the last 30 years. So what we conceptualize this entire framework is as follows. So normally, drugs aren't working, we know that. If you think about an app, and we all have our phones, we've all had the issues with either moving between Android and Apple. If you don't update to the latest operating system, it doesn't work, it's incompatible. What we're saying is actually quite simple. All of the assets are actually targeted for a hardware. They're all designed, to work on a human, a Homo sapien, which is great. What we're saying is that there's an element of variance that occurs in between, an operating system, which is the bacteria and the geographical and lifestyle approach of that individual. And this is really important because if you're within the United States or, say, in New York, those bacteria will be very different than if you live in Cambridge in the UK, which is where I'm from, or spending time, say, in India or in the Middle East. And this is one of the things from a health equality point of view. We do see there is a very large variance in how effective drugs are. But from a pharmaceutical perspective, it's imperative to know, because if you're going to run your study in Australia, as many people do, because of the attractive sort of framework from a regulatory point of view, and then move that to the US, there's actually quite a difference there. Now, from the tech point of view, the way that we've built this up is we have a phenomenally large sort of microbiome-based knowledge graph. So about 15 billion connections. So to put that in context, there was an announcement probably 3 months ago about the largest sort of biology-based knowledge graph in existence at about 5 billion. So we're at least 3 times larger than that. And, you know, it's a rubbish in, rubbish out. So it could be much larger. So that's a really tiny amount of our data. What we do is actually build up these in silico representations. So we build up the hardware, building, first of all, in silico cells. We can look at small molecules or ADCs. We can look at, you know, we can build up an individual layer, so an in silico microbiome or in silico tumor, and then actually run that phase 1 study or run that phase 2 study or the multi-center trial. Now, ultimately, this isn't a perfect system, just to say. We're not saying that we understand all of the biology, but at the moment, this is occurring in all of the pharmaceutical pipelines to some extent. And at the moment, you know, very, very few people are actually seeing this. The difference between this and AI, you know, it's important. You know, NASA, well, if you think about SpaceX and that launch, they did the self-landing rocket on its 9th attempt. Now, from a pharmaceutical perspective, you'd launch 1,000 rockets, or from an AI perspective, you'd launch 1,000 rockets and get that training data and then try and learn which aspects worked. Obviously not a feasible situation. So what we do, which is very different, which actually maps so right down to the biology, is that it's built for that mission-critical approach. You don't need training data, which means all we need is the chemical structure. So we're able to track all of large pharma. We're able to, you know, our ability to be able to look at different assets is sort of quite broad, actually. We're very computationally tight, meaning if you look at the large sort of Gen AI models, the large language models, foundational models, however you want to label them, you know, people are often burning, you know, there's the little meme about the company that had a sort of POC for 100K and sort of blew 500,000 on their ChatGPT sort of bill for the project. It's phenomenal to actually run. So if you look at how we validated this, we've actually validated this in a PK study. This was the published data. This is what our engines actually determined. The PK was from this drug-bacterial interaction in 2 approved oncology assets, and this is what the real-world measured PK was in those individuals. As a clinician, it's like, okay, that's great, but why should I care? You should care because what we then did was actually look at 4 oncology drugs and looked at the difference between the response in Japan and in the US. What you see in the blue is that there is more drug metabolism in the US population than what there is in the population in Japan. And if you look at the 1-year survival, if you live in the US, your 1-year survival is 16% lower than if you lived in Japan. And what we're saying is that there's an element of this which is caused by that drug-bacterial interaction. Not all of it, but certainly an element of it. We've then scaled this. We can now do, you know, we've looked at every single approved oncology drug. And the reason for doing all of them is we understand what degree of drug-bacterial interaction can be tolerated by a normal trial design. We're now going through the assets that have failed to see whether they land within that. From that geographical perspective and from a market perspective, looking at Japan, China, India, and the US, obviously very large markets, and this is one of our first products, is being able to run this in silico events. You know, we've published at you know, all the sort of major oncology and sort of major conferences. Our ability to be able to move at speed is that our average time from hypothesis generation to validation is actually measured in days and sometimes in hours. We have a phenomenal team. It's a very tight team, very different backgrounds from an engineering perspective. Sort of Jackie Hunt, who's the ex-CEO of Benevolent AI, is our chair with that pharma experience. We're funded by Sofinova and Hoxton. From a commercial sort of model, we have a lot of things that we can go after. We're being very focused on a bespoke, on a fixed fee and then bespoke model looking at assets. We can do this for competition as well. We have a co-development model where we can actually take a product all the way through, and we're actually looking at sort of trial rescue as well. We raised a 5 million seed in 22. We're officially partnered with Bayer. We work with the MOD in the UK, and we're part of a number of sort of startup programs. We're raising a Series A at 25 million. And thank you.